Hi, so nice to meet you, Dr. Bencourt. We've heard so much about you.
We would have loved to have met you in person, but 2020, am I right?
All right, let's get this proposal going. Hi there, I'm Trenton from Research and Development. On behalf of the team at MET Labs, we would like to warmly welcome you to our grant proposal presentation.
Hi, I'm Miranda, data analyst. We're requesting funds to sponsor the development of a melanoma treatment from an Amazonian bioluminescent plant, Green Glow Funkadelicus, using humanized mice models.
Hi, I'm Erin, our lab director. As a newer corporation, we are striving to push the boundaries of medicine by exploring new methods to stop cancer in its tracks.
We hope you see just as much value in our proposed research as we do. Our development team draws from world-renowned talent, and we are excited to put our brains together with your money to tackle the dynamic challenges that cancer presents to modern people around the globe. So let's get into it. How prevalent is melanoma? Simplifying the math a little bit, one person dies from melanoma every hour in the US, and mortality is going up with no viable treatments in sight. Skin cancer treatment in the US costs over $8.5 billion annually. Clearly, we're at a standstill in treating this fatal disease. We at MET Labs think that we have a solution. In 2019, we were all traveling together through Brazil's lower Amazon River basin and noticed a glowing green plant on a walk through the jungle. It turns out that this plant, Green Glow Funkadelicus, is highly coveted by the local Waimiri people for its healing abilities due to the very component that makes it glow. We thought, hey, if it's good enough for these fine folks, it's got to be good enough for us, and might even be good enough to tackle growing melanoma cases around the world. With the permission from our native guide, we took a small sample of G-Funk to our lab in California to begin developing a potential treatment for melanoma. After some preliminary work, we're ready to seek funding for the next phase. But first, we needed to generate a sizable, workable sample of this green fluorescent protein, GFP. Afterwards, we would be able to test it on melanoma cells to see if it would treat the disease in mice before moving on to human trials to seek FDA approval. So we designed a preliminary experiment to get there. By cloning the gene that codes for the green fluorescent protein into E. coli cells using a recombinant plasmid, then verifying the presence of the gene by amplifying it with a polymerase chain reaction, we could eventually grow enough GFP to test in mice. Now, I'll leave it to our lab director, Erin, to explain in more detail.
Thanks, Trenton. So we began our preliminary experiment by extracting the total DNA from the G-Funk leaves. Then we next performed a uniquely designed restriction digest using restriction enzymes KPN, I, and AHI to isolate the GFP gene within that DNA. We then inserted the GFP gene into a linearized PBAD vector. Then the recombinant plasmid P-Glow was transformed into E. coli bacteria and plated on selective media to grow. Some of the selective media had LB base, some had ampicillin, and some had arabinose in order to trigger the GFP gene. We then screened each plate under UV light to determine which plates the plasmid had transformed into the bacterial genome on. Once confirmed, we took samples of the transformed E. coli and isolated their DNA, and then isolated the GFP gene. Next, we used a polymerase chain reaction to amplify the GFP gene using specifically designed primers. Sequencing chromatography then confirmed the GFP genotype, and column chromatography was used to purify the GFP protein from the total E. coli protein lysate. By the end of the preliminary experiments, we had extracted pure green fluorescent protein from transformed bacterial cells. So here in this first picture, we see that the E. coli were transformed by the gene because we can see that they glow under UV. And in the second picture, we can see the resulting gel from the colony PCR run. So we can see that 3 out of the 4 E. coli colonies that were tested showed positive confirmation of transformed G-Funk DNA. We used colonies 1 and 4 in sequencing and purification, since they most closely matched the G-Funk DNA sequence for the GFP gene. Now, Trenton will explain how we used that purified GFP.
With enough GFP made and purified, we are ready to start the next phase of the treatment development, which requires funding to proceed. Our aim that we are requesting funds for is to treat humanized mice models with GFP and then test whether it reduces cancerous growth. Using mice injected with human melanoma and immune cells, we will watch the pre-melanosome protein P-mel to see if its expression levels decrease after 14 days. 3 different concentrations will be tested, and extra biopsy samples will be taken at days 0 and 7 for future research. Back to you, Erin.
Thanks, Trenton. Okay, let's enter the lab. Safety goggles, everyone. Okay, so we're gonna have a total of 24 mus musculus or common mouse models. 12 of them are going to be an experimental group and 12 of them are going to be a control group. The difference between these 2 groups is that the experimental group will have cancer, will receive that in transplant of human melanoma cells, whereas the control group will not. And we will be testing the GFP gene on both of these groups. So there's going to be 12 mice, and I have 12 mice here. What a coincidence. In the experimental group. So we're going to begin by taking the melanoma cells from the human and also the immune cells. That way we simulate the best immune environment in the mouse. We simulate a human. So we're gonna take that and we're gonna inject it in the same volume and the same concentration to all of our 12 experimental group mice. So once that is done, assuming that all of that went well and our mice live, we will then treat these mice differently. So we're gonna have 12 total mice in this large group, and then we're gonna have 4 subgroups of 3 mice each. So we've got the first 3 that receive 0 milligrams per kilogram of treatment, basically no treatment. Same thing, it is no treatment. Then we've got one group of 3 mice that has 1 milligram per kilogram dosage. We have 3 mice that have 3 milligram per kilogram dosage. And we have 3 mice, I don't know where my other mouse went, but we have 3, there we go, 3 mice that get 6 milligrams per kilogram. So we're gonna be testing what dosage is best for GFP treatment. So let me draw up the GFP. Got some nice green Gatorade for this. And here we go. So I'm using a 22 gauge needle in case that's important. And we're gonna give the mouse its GFP. There we go. Look at that. Perfect. So that was the experimental group. And now we're gonna have to do the same thing in terms of treatment to the control group. And this is so that we can make sure that the treatment isn't gonna harm healthy cells, because we want it to take out the cancer, but we don't want it to take out the mouse. So after all of that is done, we are gonna take biopsies from all of the mice. So using our needle again, we'll probably take a tissue sample, and here's little mouse number 4. We're gonna take a biopsy from him and test that later. So we're gonna take that biopsy on days 0, 7, and 14, and we're gonna create a total protein lysate from this, where we can use that in the Western blot. So here's in our diagram, here's our total protein lysate. And we're going to use that in our WB. So the next step is going to be to perform this chemiluminescent WB, Western blot, to test if the GFP injected actually does reduce P-mel expression in all of our mice samples. Oh, we're out of the lab. I can take these off All right, so in order to do that, the total protein lysate concentrations have to be determined first by a Bradford assay. So we can use equal protein concentrations in the Western blot. After we've done this Bradford assay, the proteins will then be separated and denatured with a sodium dodecyl sulfate polyacrylamide gel electrophoresis. That's a mouthful, otherwise known as SDS-PAGE. The denatured proteins will then be transferred from the gel to a nitrocellulose membrane, in which we will conduct a Ponsu red stain to confirm the success of all the previous steps. If these steps were successful, we will see red bands on each sample lane. And therefore, we can move on to immunohistochemical staining. For this immunoblot procedure, the 100 kilodalton PMEL protein will be used to quantify the effects GFP has on cancer development. This protein was chosen because it is found in pre-melanosomal vesicles and is indicative of melanocyte maturation from stage 1 melanoma to stage 2 melanoma, which is what we're looking at. This protein is also directly proportional to cancerous growth, making it a perfectly reasonable protein to analyze. The primary antibody we use to conduct the stain is called HMB45, and it's anti-PMEL. It was chosen because it reacts with approximately 90% of melanocytes that have this protein. After primary antibody blotting, a goat anti-mouse secondary antibody called HRP will be used as the labeled visualization marker. The last membrane stain will be made with a substrate that binds onto the secondary antibody and will be observed through chemiluminescence by a Western blot imaging system called the iBright FL1000. It's also important to add that a specific ladder will be chosen for this experiment. It has 8 markers that span from 20 to 150 kilodaltons in length and has the ability to function with chemiluminescent Western blots. On to you, Miranda.
Thank you, Erin. So, after this experiment is conducted, we believe that there are 2 potential outcomes most worthy of talking about. Here's what the Western blot panel might look like on day 14 of the experiment. Non-cancerous protein samples are indicated by NC, which are lanes 2, 3, 4, and 5. And cancerous protein samples are indicated by the C, the last 4 lanes. The numbers beside them are representative of the milligrams per kilogram GFP concentration injected into the experimental group of each well. The leftmost well has a molecular weight marker, and beside that, you can see what kilodalton corresponds to each ladder band. It is important to note here that the wide-banded stains at 100 kilodaltons would indicate the active presence of PMEL production in melanocytes developing from stage 1 to stage 2. It is important to remember that PMEL is 100 kilodaltons. So in outcome A, the arrows you see here pointing to the wells filled with non-cancerous protein samples are not stained at all. This means that the samples lacked a presence of protein PMEL, and in turn confirms the lack of any tumorigenesis. When looking at cancerous samples for outcome A, the ones inside the box we would see equally sized, large stains at 100 kilodaltons for all of them. If this were the result, we would be able to conclude that no amount of GFP affected the PMEL protein production, and therefore the cancer is still continuously growing within the melanocytes. This is the least desired result, as we are seeking to decrease a decrease in melanocyte development. Here's what the Western blot results would show for outcome B. Again, arrows are pointing to the non-cancerous protein samples, and like in outcome A, none of them would exhibit any staining for the same reason. Simply, there is no PMEL production in the cells sampled because they are not cancerous. Compared to the first outcome, though, we would observe a decrease in staining of cancerous protein samples from C0 that did not receive any GFP to C3 that received 3 milligrams per kilogram of GFP. In this outcome, C6 would exhibit the same amount of staining as C3. With these results, we would be able to conclude that higher GFP concentrations in cancer samples lead to decreased staining, and further suggest that GFP successfully decreased PMEL production and tumorigenesis in melanocytes. This is the optimal result. We want GFP to actually work within the melanocytes. So the outlined outcomes also suggest 2 additional possibilities, which would be part of further investigation. In these 2 images, you can see that in both outcomes, there is no decrease in staining from 3 milligrams per kilogram to 6 milligrams per kilogram. And this suggests that either more time is needed for GFP to react with the melanoma cells before a change in PML production can be observed. And in this case, a follow-up experiment would be necessary to test time as an independent variable to the GFP protein. The other option is that this could suggest that GFP has a treatment dosage threshold of 3 milligrams per kilogram, where no increase of GFP treatment concentration affects the PML production. In this case, outcome B may have reached its final PML expression levels earlier than day 14. To test this, another Western blot could be conducted from the extra 25% of the biopsied samples from day 0 and 7. Many other outcomes beside these 2 could result. For example, it is possible that we see a high expression of PML in non-cancerous cells. This could suggest tainted reagents. Another outcome could result in increased PML expression as GFP treatment concentration increases as well. This would suggest pathogenic properties of GFP. But again, these could all be further investigated. It is important to consider some possible design effects. A potential pitfall for this experiment could be the occurrence of mouse model mortality before the samples are retrieved. If this were to occur, the experiment could be repeated for shorter biopsy intervals of 0, 4, and 8 days. Another pitfall would be if the GFP proteins improperly purified in hydropathic interaction chromatography. This would lead to undesired proteins being injected into the mice models. If this occurs, alternative column substrate materials or different salt and separation buffers could be used to improve efficiency. As you can see, Dr. B, we have done all of the necessary steps to prepare for the next testing phase in our mice. Our preliminary experiments provide enough verified GFP to carry on our outlined aim.
We hope that you see the value in this proposal, as there is no better time to invest in developing a melanoma treatment from GFP. With the FDA's current COVID-19 waivers for emergency trial permits, we think we can push through a successful melanoma treatment pending human trials after our experimentation with mice. Additionally, travel right now is limited. No one's able to go to Brazil to look for any more plants. We already have it. Let's develop it.
We want to thank you so much for your time, attention, and consideration. We're looking forward to working with you, and we hope you enjoy your holiday.
Bye now.
Bye now.